Today I will be discussing a neurological disease called dementia through an extensive summary and analysis of how iron metabolism is associated with the aging brain. Dementia is the term to describe a collection of symptoms that arise as a result of various disorders affecting the brain. Neurodegenerative diseases typically present themselves in aging and characterize a progressive deterioration of the nervous system. If left untreated, chronic stages of dementia result in Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis, and Huntington's disease. Dementia is currently the second largest cause of death and continues to devastate Australia's health economy each year. Worldwide, it affects a whopping 50 million people, with approximately 10 million new cases being diagnosed each year. Because this disease affects the aging population, it is important that we focus our efforts to deal with it early on, as statistics show that without a medical breakthrough, it is expected that the cases of dementia will almost double over the next 30 years. The tissue of the brain is made up of high concentrations of endogenous iron, zinc, and copper. Iron, being the most abundant metal ion in the body, is highly essential in the process of oxygen transport, oxidative phosphorylation, production of myelin, and regulation of neurotransmitters in various cells and proteins. Although relative amounts of iron are essential, recent studies have discovered that the regulation and metabolism of iron inside the brain becomes more difficult with old age. This is evident now as modern experiments have traced the influx of iron consumed by food into the brain, and when compared to other organs and tissues of the body, its accumulation was far greater than that of any other tissue or organ inside the body. A meta-review by Crichton et al. Discovered the deterioration of the blood-brain barrier due to age and other pathophysiological factors. These types of reviews encourage future analyses of the brain composition, where strong correlations were found between the accumulation of iron in the brain to neurological decline. An MRI scan performed in 2012 experiment analyzed iron amounts in the basal ganglia of 143 subjects of mean ages 11, 70, and 72 years. The study ultimately found correlations between excess iron deposits in certain regions of the brain to a lower cognitive ability. Additionally, an analysis of the experiment by Rao 2010 revealed associations of higher iron concentrations in both the caudate nucleus and putamen with longer reaction times and reduced scores on the dementia rating scale. Vascular dementia is a prominent subcategory of dementia, alongside Alzheimer's. Although the exact nature of the pathology is unknown, recent studies on rats with vascular dementia and results from an experimental model discovered that oxidative stress, inflammation, and apoptosis were the main causes. These specific symptoms have been heavily correlated to iron deposition within the hippocampus. Furthermore, a systematic study by Wan et al. Discovered autophagy dysregulation to be correlated with high levels of iron accumulation in the brain. Autophagy is the homeostatic process which mediates removal of ubiquitinated and aggregated cells that cause synaptic impairment, damage to organelles, and cell death if not removed. The failure to maintain autophagic reflux results in an imbalance between autophagosome formation and degradation. The study had ultimately concluded that the accumulation of autophagosomes were a primary cause of Alzheimer's, as well as other neurodegenerative diseases. Additionally, a systematic review by Aiton et al. Discussed several experiments which found links between an excess iron in the body to cognitive decline and dementia. It gathered that two studies which assessed how the composition of iron would affect cognitive ability in two separate groups, one with normal cognitive functioning and the other with minor cognitive impairment. A relationship was found between those with higher levels of transferrin, a glycoprotein mediating iron transport, with a higher risk and precursors to Alzheimer's. In the other cohort, those who had a higher concentration in cerebrospinal ferritin were found to have accelerated levels of cognitive decline. Ultimately, the study gave indication that vascular permeability was a primary cause of higher iron deposits in the brain. A very recent study in 2019 discovered the formation of beta amyloid plaques and tau neurofibrillary tangles associated with Alzheimer's disease. Under physiological conditions, formation of such plaques form toxic oligomers and fibrils and rapidly aggregate. As a result, oxidative stress occurs which then cause neuron degradation. The detection of amyloid plaques is now a commonly known indicator for the identification of Alzheimer's disease. In summary, current systematic reviews and current evidence which link excess iron deposits to cognitive decline are still very much in research, as data is still very much sparse and provide a limited field of view. More work needs to be done in order to evaluate the links between cognitive function with varying levels of iron intake and brain deposition. Areas that require more extensive research include examining how iron deposits in different locations within the brain affect cognition. This includes a greater usage of biomarkers to provide a more accurate picture of iron homeostasis, so that various iron types and storage mechanisms can be better understood. More follow-up experiments with more complex MRI scans would improve the breadth and sensitivity of results, while more extensive epidemiological studies that include a larger population and longer time frame can help us uncover the potential pathways of neurological disorders.